It is 11 o'clock Moscow time, and we are going to start our first invited lecture at the course of Model and Methods of Quantitative Genetics. It is my pleasure to introduce and announce our first invited speaker, Professor Gordan Lauc. Gordan is a professor of biochemistry at the Department of Pharmacy at the University of Zagreb, and he is also many other things, but among them, he is also a founder and CEO of Genus Glycoscience. This company has analyzed more human glycomes than the rest of the world combined. Obviously, Gordan and his team are at the cutting edge of research in human glycomics. Among other things, Gordan is a co-director of the Human Glycome Project. Last but not the least, Gordan is a very good friend and a long-time collaborator. I think by now we collaborate close to 10 years, and this was very fruitful. It was always a pleasure, and it was a privilege to work with you, Gordan. Among the projects we offer to the students of the course, two concern glycomics. Without further ado, I pass the word to Gordan. Gordan, please, the floor is yours.
So thank you, Yuri. So thank you for the introduction. I will try to tell you something about the Human Glycome Project and what did we learn from the analysis of the first 150,000 people. Yuri already mentioned that I am the founder of the Genus Glycoscience Research Laboratory. I also own a company called Glycomics, and I do have a couple of patents in the field, so I'm not completely unbiased about the field that I'm talking about. But before I start with glycans, I would like to bring you a bit backwards and talk about personalized medicine and the medicine in general. And one of the big problems we have in medicine today is that we treat all people as equal. We think they're just a Homo sapiens. And then we give them some kind of a therapeutic approach, and it generally works only in 20 to 30% of people. In 80% of people, this is a failure. And then we try another one, and then we try another one. And this is the way medicine works today. Personalized medicine, the idea that we can have some kind of a biomarker panel which would stratify people into groups and then give them ideal treatment, is still the holy grail. We are all trying to find it, but there is very limited application of real personalized medicine in today's medicine. And one of the reasons is that people were mostly focusing on genetic information. And despite the technical success of the Human Genome Project, the genetic research did not actually yet fulfill expectations of science and general public. There is very little translation of genetic data into actual clinical work. There is some in pharmacogenomics, a little bit in oncology, but still information about our genes is still not helping making clinical decisions. And one of the reasons for that is that simply genes do not define everything. And when we talk about complex diseases, only about 30% of them are genetically defined. And when you talk about longevity, it's only about 20% genetic. And I think one of the best examples are these two pairs of identical twins, which have every single letter in their genomes identical, but they obviously have a very different life trajectories. And one of the reasons for that is that genes and genetic polymorphisms are very far away from the final phenotype. So when you start from some kind of a genetic polymorphism, and then you make RNA, and it's being processed, and there are some products, and there are some proteins, and then proteins interact with the metabolites, with the environment. There are many layers of complexity that are between a genetic polymorphism and a final phenotype. And it is very difficult to make these connections. And usually when there is a connection, effects are relatively small. So this is quite difficult to translate into a clinical decision. And one big part of the picture which is missing are the glycans. I like to think about glycans as the ultimate layer of molecular complexity. But in the same time, they're the most neglected molecules of cellular communication. And one of the best examples of that happened in this COVID pandemics, when even the director of NIH was tweeting about a protein called S-glycoprotein of SARS-CoV-2 without a single glycan attached to its structure. Well, actually, the protein looks like this. There is more glycan on its structure than actually polypeptide. And these glycans are not only decorations, they are part of the molecule, they participate in interaction with the receptor, they determine molecular stability, and they're active participants in at least SARS-CoV-2 infection. And it's not only SARS-CoV-2, it's not only S-glycoprotein, the majority of proteins are glycosylated. And these glycans are important structural component of a protein. So if we look at these two proteins, and the gray is a polypeptide, and these colored parts are glycans. Glycans are integral part of the structure. So you cannot understand the structure without knowing the glycan part of the structure. And the analogy I like to make is that protein without its glycan is like a bird without its feathers. So we can do a lot of research on these birds. We can study bird physiology, we can study bird biochemistry, but we cannot see this bird flying because bird without its feathers cannot fly. The same thing is for a protein. We can study this polypeptide part, but we cannot understand function of the entire protein without knowing the glycan part of the structure. And one of the big features of the glycan part is that contrary to polypeptide, which is encoded by a single gene, so there is a gene which determines structure of a protein, glycans are encoded in a complex network of genes. So there are dozens, maybe even hundreds of genes which work together, and this is a network for IgG, which determines which will be the final structure of a glycan, which will be added to a specific protein. So glycans are encoded in a complex dynamic network of hundreds of genes. There is a biosynthetic pathway with many enzymes which build a final glycan, but all these enzymes are being regulated in some way. And there are some other proteins which contribute to this biosynthesis. So there is a whole network of genes and proteins working together to make a glycan. Unfortunately, the majority of research today is being done on genomics and epigenomics, which is actually a very myopic view because genes, and even with epigenetics on top of it, it's a very low information capacity. Much more biological information is stored in proteome, lipidome, glycome, where there is a lot of information, a lot of structural complexity. Just as an example, if we think about nucleic acids have only 4 building blocks, 4 blocks which determine the complexity of the structure of nucleic acids. Then the second layer of complexity are proteins. With the 20 different amino acids, they can form many more different structures than what nucleic acids can do. But the glycoproteins are actually composed of over 2,000 different glycan blocks. So parts of the glycan which are assembled kind of Lego blocks. And then the complexity of glycoproteins is way, way larger than of the proteins. And the glycoproteome, so the entire set of different glycoproteins, is several orders of magnitude more complex than the proteome. And these glycans are actually responsible for a large part of inter-individual variations. So the reason why we are all different is by large defined by glycans. And even the first of example of molecular differences between the people, the A, B, and O blood groups, are chemically glycans. So these are O, A, and B antigens, which are the basis of the ABO blood groups, for which the Karl Landsteiner got the Nobel Prize in 1930. And this is not something which only I am claiming. There are very strong policy documents, both in Europe and in US, which claim that glycans are directly involved in the pathophysiology of every major disease, and that the additional knowledge from glycoscience will be needed to realize the goals of personalized medicine and to take advantage of the substantial investments in the human genome and proteome research and its impact on human health. So these are the policy documents which are now more or less accepted, but still the large number of people, unfortunately, ignore glycans, which is not good. And I think one of the best examples are monoclonal antibodies. So 8 out of the 10 best-selling drugs in Europe today are immunoglobulins. Immunoglobulins are by large determined, their function is by large determined with the glycans attached to the Fc domain. And I think probably one of the best examples how important this is are 2 monoclonals, rituximab, which is on the market for already over 10 years, and the next generation of the same anti-CD20 antibody, which is ublituximab. The only difference is that ublituximab does not have this core fucose attached to their glycans. And ublituximab is effective in 100 to even 500 times lower concentration, because this core fucose is a safety switch which is actually preventing rituximab from doing its purpose. So by just understanding that we have to make monoclonals without this core fucose, we can make a drug which would be probably 100 times cheaper just to produce it. But this glycomics, the glycan analysis, is something which is actually not easy to do from a number of reasons. One of them is that glycans are non-linear, complex chemical structures which cannot be amplified in a lab. So there's no something like a PCR for glycans where you can take a small trace of a DNA and amplify it in a large amount. For glycans, we have to work with the biological material we isolate from nature. And then we have to do many different chemical steps to isolate the protein, release the glycans, label the glycans, so that we can actually separate them using one of the techniques and then get the final readout. And this is particularly difficult in high throughput. So doing this in a way that is reliable enough to analyze thousands or tens of thousands of people is something which is not easy. And there is actually only a very limited number of labs in the world which can do it. The one which does the most is my lab in Zagreb. The second largest lab is in Leiden University Medical Center. And there are some attempts in some other labs which either actually shut down or do not operate in this field at the moment. So one of the reasons why we established the Human Glycome Project is to help people to use this glycan analysis technology to define structures, to understand functions of human glycoconjugates. The whole story started a little bit more than 3 years ago, where we met for another conference in Dubrovnik, in Croatia. And at one point we said, wow, why don't we start the Human Glycome Project? Let's start to collaborate and work together. And at the moment, the Human Glycome Project has over 44 individual projects which contribute to this global goal. And we have over 300 participating researchers which collaborate to resolve these key questions. And we have a number of divisions of the Human Glycome Project. I'm leading the division which is focused on inter-individual variability of the human glycome. And this is something that I will be mostly talking about today. So far, we analyzed over 150,000 individual glycomes. And these glycomes were coming from some of the best genotype and phenotype cohorts in the world. including, for example, the Twins UK cohort of over 10,000 twins, where we analyzed over 20,000 individual glycomes, and many other epidemiological and clinical cohorts, which we did in collaboration with some of the best researchers in the world, including Yuri, of course. And this enabled us to really shift glycomics toward the mainstream science. Because a couple of years ago, it was really difficult to publish a glycomic paper in a leading journal. But now the situation has changed, and adding good glycomic data to your clinical data set is actually helping you to get into the best journals, because we are now routinely publishing in the best journals in the field, like gastroenterology, circulation research, diabetes care, and so on. So really, glycomic information can help you understand what are you working on and better understand biology of your problem. And fortunately, we managed to finish this first big fishing expedition, which is in science often considered like something negative, meaning you are just collecting information without using your brain too much. I got the message, media cannot reach the server. Can you still hear me?
Yes.
Everything okay?
I can hear you.
Okay, there was an error message on my screen. I still have the message, media cannot reach the server. Okay, I will just continue. So we did this initial acquisition of information, so-called big fishing expedition, and now we have a huge set of information which we are using to mine the gold and trying to understand how these things actually work. And of course, one of the first thing is to try to understand genetics, protein glycosylation. And this is this long-lasting collaboration we have with Yuri and his team. We have a number of publications just on IgG glycosylation. We have already 5 GWAS papers, which mapped the network of over 40 genes, which we know now, not all published yet, which work together to make a decision about a glycan structure, which is being added to immunoglobulins. So what we actually see here, we see that the glycoprotein is a biological structure or a chemical structure that is being inherited as a complex trait. And when we think about alternative glycosylation, so putting different glycans to the same position on a glycoprotein, effects of alternative glycosylation is functionally equivalent to coding mutations. So we know that if there is a coding mutation, if the amino acid is changed, the consequences could be lethal. So a single mutation could kill you. Here, we have a chemically similar consequences. The protein is different. We have different glycan attached to its structure, but this is not determined by a single gene. This is being inherited as a complex trait affected by multiple genes, but not only genes, also epigenetic regulation of these genes and the environment. And one of the interesting studies we did was by looking a collaborative cohort of mice, which were established in the University of Western Australia by the Grant Moorhan. So he started with the 8 founder strains, interbred them for 3 generations, and then inbred them in 100 plus strains of cousins. So these individual mouse strains were kind of a cousins, all originating from 8 grandparents. And in these 3 generations of reshuffling the genes, these mice became so different in IgG glycosylation that we could actually stratify them in the different groups just based on IgG glycosylation. So we saw something which looks like a microevolution. By just reshuffling genes, a novel chemical structures appeared, or at least novel compositions appeared, which made these individual strains of mice different. And this is really exciting because this is kind of an evolutionary tool which can improve changes which we need for evolution. And this is not only genetic, and this is not fixed in a way genes are fixed. So our genes are fixed for a lifetime, they don't change, but glycosylation part is dynamic. And for example, for IgG, we know that when we are young, we have glycans attached to immunoglobulins, which are kind of anti-inflammatory. They are suppressing inflammation. And as we are getting older in our biological age, our glycans are becoming more pro-inflammatory, and they're actually promoting inflammation. And when you think about the cell surface, and this is the cells under the microscope, these are the cells. This tiny white thing, this is the lipid bilayer, and these thick things on the surface of the cells are actually glycans. So glycans on the surface of a cell are protective walls, which are protecting the cell from anything coming from outside of the cell. And this is done by integrating genetic, epigenetic, and environmental information. So there could be genetic polymorphisms, which have a specific consequence on a glycan. Very similar thing could be achieved by epigenetically downregulating the same gene, but also there is direct environmental factors, including the diet or the hormones or whatever, which could have the same effect or compensate. So integration of genetic, epigenetic, and environmental information makes a decision about which glycan we are going to put at a specific site and what are going to be a functional consequences. So if we talk about immunoglobulins, they have two key functional components, FAB part, which binds to antigen, and FC part, which activates effector functions. So this part decides what will happen after we bind to antigen. Because don't forget, only a small fraction of antigens are actually dangerous. Most antigens we are seeing is food, something kind of airborne, and we should actually ignore them and not activate the immune response. And this is something which is determined by these glycans. Here, for example, if there is something we call core fucose, so fucose attached to this first N-acetylglucosamine, this is blocking binding to the Fc gamma receptor 3A, which is blocking activation of antibody-dependent cellular cytotoxicity. So the NK cells don't kill the target if it's being recognized by an antibody with the core fucose. You remove the core fucose, and the target is being eliminated. Another aspect, for example, if immunoglobulins and with these N-acetylglucosamines here, they're actively promoting inflammation. They're activating complement, they're activating the lectin pathway, and they're destroying the target. But if glycans are extended with galactose and sialic acid, these immunoglobulins actually suppress inflammation. So they start to dampen the inflammation, and they're used as a tool to suppress too much inflammation. And there is a lot of research about it. I think pharma industry is investing hundreds of millions of euros to try to understand how glycans modulate activity of the monoclonals, which are the best selling drugs at the moment. Something what we are focusing on is how different people are in IgG glycosylation. And if we look at this figure, this is 6 random individuals with a different glycan presented just as a different color. And just by looking, we can say, yes, people are different. Person A has different IgG glycan composition than person B. So what determines these differences? Approximately, or on average, 50% of it is genetic. So genes which we have determine approximately 50, on average, 50% of heritability. For some glycans, it will be more heritable. Some other glycans, this would be less heritable. So on average, half genes, and the other half is environment. And one of the first environmental factors we identified as an importance for IgG glycome was the age. So we were looking in our population cohorts, And these here are two Croatian islands. These are twins from UK. These are two Croatian islands. This is the Orkney Islands in Scotland. Blue are men, red are women. Change with age, different glycans. We see that they change significantly with age. And they change in a way which I mentioned before, that the structures which are suppressing inflammation are going down with age. And actually, these structures suppress inflammation. And the structures down here which promote inflammation, they go up with age. And if you combine that, only this information here, what happens to glycans, you can actually predict somebody's chronological age, not extremely accurately. It's plus minus 9 years. For example, DNA methylation is way more predictive in guessing somebody's chronological age. I think on average, the error is 1 year. But what is interesting, that the difference between the age that the glycans predict, so-called glycan age, and the real chronological age can be explained with a healthy and unhealthy lifestyle. For example, we see that after correction for chronological age, glycans and the glycan age associate with the biomarkers which we know are associated with unhealthy lifestyle, like high insulin, high fibrinogen, HbA1c, BMI, and so on. Also, when we looked at the different cohorts of people with different diseases, so each column here is a different cohort. Last four are just a general population cohorts, and the effects of age are here shown. So what is the difference between old and young people? And we see that in many different diseases, and this is lupus, here colitis, Crohn's disease, rheumatoid arthritis, Hashimoto, type 2 diabetes, colorectal cancer, most of these diseases make people look older. So their immunoglobulins change in a direction of accelerated aging. So what we see here is that glycans are not a specific biomarker for a given disease, but they change in the same way in many different diseases. And the way we currently think how we can explain this is that these glycans actually regulate low-grade chronic inflammation. And by regulating low-grade chronic inflammation, they either contribute to disease development or suppress development of many different chronic diseases. Another thing we did is we looked all around the world and asked our friends to send us samples from people in their part of the world. And we collected 27 populations. We had 100 people in each of them. We had as wide as possible age span, fully randomized across 32, 96 well plates, did glycan analysis, and saw that people from different countries look different. So depending where you live, your glycans would be a little bit different than if you lived in another part of the world. And then when we looked what explains these differences actually most strongly associate with the Human Development Index, which means how developed the country is where you live, but also it's very strongly correlated with the expected lifespan. So if you live in a country where your lifespan is expected to be higher, your glycans move to old IgG glycome slowly than if you live in a country where you are expected to live shorter. This is a very intriguing concept. It's very hard to decide anything about causality, but at least we know these two things are associated. So now we try to think, how can this happen? What is regulating all this process? And then I come back to this network of genes, which we mapped to the series of GWAS papers with thousands and thousands of people who were involved in this study. And then we see that these genes group with some of the enzymes which build glycans. So these could be either regulatory genes for these enzymes, or they could somehow act together to modify glycans in this specific way. So one of the ways which we are trying to confirm is, are these genes actually regulatory? So would change in expression of this gene here, for example, change the expression of glycosyltransferase, because this is one of the simplest way how we could imagine these genes could be working. And for some genes, we do see, yes, they regulate glycosyltransferases. Most probably, some of them are direct transcription regulators. Some other act more complicated. But some of these genes, we don't know how they work. For example, they're not even the B cell genes, so they're not expressed in the cells where immunoglobulins are being made. So their effects have to be way more complicated. Another interesting thing which we learned is that these genes, which regulate IgG glycosylation, show very high pleiotropy with a number of other different phenotypes, including some inflammatory diseases like lupus or Crohn's disease, some autoimmune diseases, but also some neurological diseases like dementia or Parkinson, or even cholesterol level or HDL level. So these genes which regulate IgG glycosylation are known risk factors for many different things. Of course, this does not mean that the causality goes from a gene to a glycan and to a trait, but for at least some of them, it may be going in this direction. And then we can try to identify them and see how do they work. So one of the first thing is to look whether genes are altered in some of diseases. And we just published a review, it's already nearly 2 years ago, listing over 300 studies showing that glycans change in a different way in the different diseases. And one of the examples I will present here is cardiovascular diseases, where we know that glycans change in people with hypertension, but they also move in the same direction when people have only pre-hypertension, so they're not fully ill yet. And there was a couple of papers by Phil Scholl from UT Southwestern, which suggested that it's IgG glycosylation that could be functionally important for the development of hypertension. He even he identified a pathway through the Fc gamma receptor 2B, how this could actually be working. So we teamed up with him for the latest paper, where we were able to show, actually where he was able to show in mice, that by feeding mice with N-acetylmonosamine, which is a precursor for sialic acid, you can prevent hypertension in obese mice. So you put mice on a high-fat diet, they develop hypertension. You put them on the same high-fat diet, they become fat. But if they have MANAC supplementation, they do not develop hypertension. And we, in parallel, we did a study on humans, but not intervention. We just did a correlation. And we saw that indeed, in people with hypertension, we see less cellulation than in people without hypertension. So this actually may be a causal mechanism how glycans lead to hypertension. When we looked in the population cohorts, we saw that IgG glycan composition today strongly associates with the cardiovascular disease risk score. So what is the risk to get cardiovascular diseases, which is calculated by the people who do this type of calculations? And this was a study on over 3,500 people in Scotland, showing that indeed the glycans associate with the risk. Then using this data, we managed to persuade people from EPIC to give us access to their samples. So the EPIC is a large study in Europe where over 300,000 people were collected nearly 30 years ago. We got access to the German subset of 27,000 people. And out of these 27,000 people in a 10-year follow-up, there was 508 cases of cardiovascular events. So people either got a heart attack or stroke, and the samples were collected before this happened. We looked into glycans, and glycans and age, if combined information from IgG glycan composition and just age of a patient, was as predictive as the entire AHA score, which is currently the score used for prediction of cardiovascular diseases. So IgG glycome composition has all this information stored in it. And actually in women, only a single glycan plus age, this structure here, plus age was more predictive than the entire AHA score. So what seems here that indeed glycans attached to IgG have some kind of predictive power what will happen in the future. And based on that, we developed this glycan age test, which we think is a kind of a test. We call it biological age at the moment, but the idea is that it's actually measuring how healthy you are at a given moment by quantifying molecules, these here, which actually do the damage. And in the same time, they're based on both genetic, epigenetic, and environmental information. Because since this is not only genetic, the question is, can we change our glycans? Is there something we can do to improve our glycans and decrease the risk of diseases? And we will have many people who have been tracked for a long time. And one of these people is me. So I'm tracking my IgG glycome for over 5 years now. And my glycan age is lousy. I'm over 70. And it's getting worse. Beside here and here. What happened in these two events, I actually decided to live healthier. So I ate less, I exercised more, I lost 10 kilos, and then my glycan age starts to go back. And it doesn't have to be only weight. There are four key components that affect the IgG glycome. One is, of course, diet and physical activity as a second, but also sleep and stress. So my choice was always first, let's try to sleep more, let's try to be a little bit less under stress. Did not work out for me well. Even when I tried to exercise a lot, did not work well for me. Unfortunately, my only way to improve my glycan age is this one here. So I have to be on a diet. All other parameters are not so important. So I put this hungry monkey, which I will mention later in my talk. This poor monkey is on dietary restriction for 20 years. So it is healthier, but he does not seem to be very happy. Some other people might have more luck. Maybe it's only sleep for them or stress or maybe a little bit more activity. So the thing is, with this tool, you can actually measure and see what is working for you. And we are trying to find some kind of a general patterns. So one of the things which we learned is indeed it's obesity, which is one of the main drivers. And we did a cohort of people undergoing bariatric surgery. Bariatric surgery is removing part of your stomach. You eat less, you lose kilos, and your glycans improved. We did the same thing by looking at 2,000 twins in 3 time points. Those who were gaining weight were aging faster than those who were losing weight. So depending whether you gain or lose weight, you can change your trajectory of this glycan aging. But there does not seem to be a magic diet. We compared 5 different diets, and they help some people, they don't help other people. So everybody has to find a magic solution for themselves. And the same thing for exercise. Some types of exercise help some people, other type of exercise help other people. There is no standard human, and more or less everything you do has to be tailor-made for yourself. There are also some kind of anti-aging approaches which seem to be working. There are different biohacking approaches, and at least these 2 guys, Joseph Raphael and Chris Gatton, they are kind of gurus working with their followers, and most of their patients or followers are 20 to 40 years younger. So these things seem to be working. Something where we did the proper science is that estrogen is working. So if there is a suppression of estrogen, and there was a study on ladies where menopause was chemically induced, glycan age goes up approximately 30% in all of them. If there is estrogen supplementation, this does not happen. After recovery, they go back to normal. So there are approaches how you can actually change your glycans. And I would like to remind you, they're actually the effectors. They do the work. So if you change them, things will change. And for immunoglobulins, it's regulating inflammation on multiple levels, and kind of general idea, the big picture is Normally, people go to hospital when they are ill, when something hurts, when some of the organs is not functioning. It's usually point of no return. They got the label of a chronic disease, like Crohn or lupus or whatever, and they stay ill for the rest of their life. They can get treated, but they never get cured. If we find them early, if we find them while only the molecular biomarkers show there's something going wrong, lifestyle intervention can put them back in the normal healthy living. The thing is, we know that, people know that. There was a very convincing study showing that if you live a healthy life, you will gain 10 years of healthy life to your lifespan, but we don't do it. We don't do it because it's difficult, and not eating or exercising today to be healthy in 30 years is not too attractive. We don't see the feedback soon enough. And I think the most famous example are these two monkeys. This one is on caloric restriction for 20 years. This one is not. This one looks much older, but this one doesn't look much happier. Even he might probably live a few years more, but if you're living all your life hungry, what's the big deal? So the idea is that what we try to do with this glycan age is give people feedback, give them objective quantification of what is the current state, hoping that by knowing now that what I did 3 months ago was good, people will think more about doing something good for themselves. And this was one part of my story, and I see I have only a few more minutes, but I will just tell you another story really quickly, and this is diabetes. We know that glycans are active participants in the development of diabetes. There are a number of papers showing molecular mechanisms how this happened. And we bumped into one of them when we were doing our first GWAS. We published the first GWAS of the glycome 10 years ago, which identified HNF1 alpha as the key regulator of glycosylation. And this HNF1 alpha, if you have a single non-functional copy of this gene, you will develop a maturity onset diabetes of the young. So it's a autosomally dominant mutation which is causing diabetes. And then we showed that this fucose can be used as a biomarker. You can identify this small subgroup of patients with this monogenic diabetes. But then the question was, if the mutation in a gene is causing a disease, would epigenetic silencing of a gene, so this is something wrongly, so can epigenetic silencing have the same effect on the patients? And we managed to show that these changes are actually functional. So if you do epigenetic silencing of this gene, it will change glycosylation. And in a small cohort of patients, we did manage to show that some of these people who have type 2 diabetes will have HNF1 alpha in their pancreatic islets inactivated by methylation completely, some of them partly. And then we went to the big population cohorts. We saw that people with diabetes look different than people without diabetes. But interestingly, this was a prospective cohort. These differences are visible even 5 to 10 years before people develop diabetes. Went back to the EPIC cohort. This EPIC cohort had also 800 cases of diabetes, checked how predictive glycans are, and again, glycans are more predictive for the future development of diabetes than any other biomarkers which are currently available. And here I would like to remind you that these glycans are not only biomarkers, they're functional effectors in some of the types of diabetes. And I think we have identified the molecular subtype of diabetes. This is people who develop hyperglycemia in acute disease. So if there's acute inflammation, some people will develop hyperglycemia, some will not. Those who will most probably develop also diabetes in the next 4 to 5 years, and we can use glycans to practically nearly completely identify them. So at the very end of my talk, I would like to just summarize this glycan biomarker story. So we have these changes in the glycome, which we call the Diabrisk test, which are visible a couple of years, 5 to 10 years before any other symptoms. So glucose is still normal, HbA1c is normal, but glycans have changed. it seems that it's only a subgroup of future diabetes patients which have this risk factor. And we think that these glycans are not only a biomarker, but the active effector in the disease development. And as you know, diabetes is not a single disease. It's many different molecular pathways leading to one phenotype, which is altered glucose. And we are currently trying to look whether you can, if you improve glycans, will you decrease the risk of diabetes? And at the very end, all we did is very collaborative. We collaborate with dozens and dozens of institutions all around the world. We are very generously funded from numerous research projects, mostly Europeans. We got over 15 million euros for glycan analysis in the last couple of years. And I manage a reasonably large research group, and all these people work in a lab, so I can not travel around anymore, but at least I can talk to different people all around the world. And I thank you for your attention. I'm now finished with my talk.
Thank you very much, Gordan. Really interesting talk. I'm following the story very closely. Also for me, it was very interesting. Now we are open for questions, and I invite every participant who has a question to either send to the chat, or you can also send to the chat, like, please give me the microphone, and then we can pass you the microphone. I would like to start with a question from Sotpal, who is asking which part of the glycome science is the most interesting for pharma. Is it, for example, like an engineering of antibodies, or could you comment on this?
Yes, so at the moment, the topic which pharma is most heavily investing in is glycoengineering of antibodies. So they are trying to find a way to make more antibodies with the proper glycome and which has a defined glycosylation. Because one of the big problems pharma has, once they register a drug, when they get approval, this approval also defines the glycome composition which drug has to have. And there are some examples in the past where companies would make a new production facility, they make a larger bioreactors, and then they cannot reproduce the glycosylation pattern from the original drug. So they cannot use the new facility. So it's understanding the way glycosylation is regulated, how it's being affected by the conditions in the bioreactors. This is something they work really heavily on.
Thank you, Gordan. There is next question from Alexandra. Is there anything close to mutational signatures in cancer genomics for glycomics? Any strong associations which could be used as biomarker?
Yeah, this is something people were trying for decades. There are some, like for example, usually mucins have much shorter glycosylation, so there are some examples of glycans which are kind of ubiquitous for different tumors. But in general, for cells, not so successful. And the reason why I think it is not successful is that glycosylation machinery is so intricate and so complex that once the cell turns into a tumor, there are so many changes, because tumor is not a well-defined cell. All cells are different clones, different changes. They can lose chromosomes. They can do all type of mutations and rearrangements. And then the glycome is so aberrant that you cannot really see anything logical there. So I make an analogy. It's like when you have an avalanche, all this snow falling from the mountain and going through the village, and then you try to look what is left of the village. You don't see anything because the avalanche destroyed it all. So I think that for glycosylation, tumor is an avalanche. which makes the total chaos. But there are some attempts. People, for example, now try to combine anti-cancer drugs with the glycosylation limiting drugs. And there are some examples that by also adding changes in, by limiting glycosylation, you have better success with some other anti-cancer drugs.
My understanding is also, but yeah, you correct me because you definitely know more about that, that also glycosylation is thought to be quite important in metastasis because that's essential part of the cell-cell interactions.
Yes, so there are some requisites for metastasis. So some glycans are needed for cell to actually home somewhere else. But nothing is so tumor specific that you can say, okay, this is the way I'm going to make my drug, because the same mechanism would be used for homing of, I don't know, lymphocytes or some other cells moving. So it's, there is still no magic bullet against glycans, which would really help in tumors.
Yeah, clear. I think there are a few people still typing, so there may be more questions coming, and while they are coming, maybe I can ask my question. I really like this story with ciliation and supplementation in mice, where you supplement mice with MNAC, and they are still developing obesity because of diet. They are kind of healthy obese. So this is like, well, pretty cool. So what are the perspectives? Like, yeah, cannot we just add some MAC in our diet and, be as healthy as your mice?
So there was actually a clinical trial on NIH where they were feeding people MNAC. And we got the samples, and the MNAC did not change IgG glycosylation in humans at all. Now, there are two options. One is that we have more complicated regulation of IgG glycosylation. And the second one is that in mice, it actually works in obese mice to improve on decreased cellulation, while we were testing only healthy people. So I think something what Phil in UT Southwestern, who was leading that paper, is now planning to do is to try to feed obese people MNAC to see whether it will improve. So yes, of course, we would all like to have a magic pill which will enable us to eat a lot, be fat, and be happy and healthy. We don't have it yet.
Yeah, obviously we would like to, yes. Right. I don't really see more questions coming. I actually have some, but that's, oh yeah, here. A question from Kirill. You've told about glycan analysis is carried out in only a few labs, in several labs. How soon can these methods start being used in industrial practice for disease prediction, for example?
Okay.
I think it's like, outside of scientific domain, how quickly.
So I was maybe not completely clear. So it is high throughput glycomics, which is being done in a limited number of labs. There is, in a recent study where we were comparing different labs, for glycan analysis, there were over 100 participating labs. So there is over 100 labs in the world which can analyze glycans in a low throughput. But this is not answer to the question. The question is, when can this be used in diagnostics? This is one of the key goals of the Human Glycome Project, because to have something registered as a diagnostic test, it has to be performed on a machine which has been certified as a diagnostic machine for this type of an assay. For glycans, we don't have it at the moment. So we are talking to the instrument providers that they certify some of the machines for glycan analysis. And personally, I'm most optimistic about the capillary electrophoresis, because all those old DNA sequencers which were using capillary electrophoresis can easily be repurposed for glycan analysis. It's only the software which is needed. All the hardware can stay the same. So we are currently trying, for example, for this glycan age, we are trying to develop a solution where it could actually be done on a DNA sequencer, and then in principle it can be done in any lab which has a DNA sequencers, and there are many in the world. So I hope not too far away from now.
All right. Well, thank you very much, Gordan. Thanks everyone for the questions and for being here. We are actually very well on time with the presentation and with the lecture. So, okay. Oh, we have one more question, I see. Well, I think it's not a question, it's a statement. We still have last question from Evgeny. Thanks you for the presentation, Gordan. And which technologies can give the highest resolution on plasma glycosylated proteins regardless to price? So basically, I think the question is like, if you had all the money in the world, what would you do?
Yeah, this is a difficult question. So currently there are more or less 3 technologies which are being developed. One is mass spectrometry, which is for the plasma would most probably be combined with chromatography at the front end, so liquid chromatography, mass spectrometry. And the second is just chromatography with the fluorescent labeling, and the third one is capillary electrophoresis. So we use all of them. The problems with the mass specs is that they're not so reliable machines. You need a lot of experience. It's very hard. They're not robust enough. And I think the question is, what are you looking at? Because, for example, when we talk about diabetes, for diabetes, we just look at the kind of global change in branching, and we don't need very high sensitivity. I think this can even be moved to some kind of a nanofluidic device, and easy assay can be developed. Hasn't been done yet. If you want to look at a very specific glycan on a very specific protein, I think the biggest challenge here is actually to fish out that protein, because the glycan exists also on other proteins. So I don't have a universal answer to that question, but something which is being developed mostly at the moment is mass spectrometry. So maybe if you want a single technology, it would be mass spectrometry.
All right. Thank you very much, Gordan. Thanks, everyone. With this, I'm closing this first invited talk of this course. So thanks a lot, everyone, and bye-bye.
Thank you, Yuri. Bye-bye.